The next type of anchoring junction we are going to talk about are the Desmos Ohms. Desmos Ohms are intracellular button-like welds that basically you can think of these Desmos Ohms like titch button on two pieces of cloth. They come together when you apply a little force, the titch buttons stick together. So that is basically sort of the analogy I would like to give regarding the Desmos Ohms. So internally, they serve as anchoring sites for rope-like intermediate filaments. Externally, these Desmos Ohms, they have sticky molecules. In this case, the sticky molecule is again a cadherin molecule. It's a special type of cadherin molecule. We'll talk about this particular cadherin in more detail in a little bit. But I just want to mention here, first of all, that these molecules responsible for this attachment are again cadherin molecules, and they come together. There are other proteins that assemble on the cytoplasmic face of this particular cell, forming this dense plaque, which is referred to as Desmos Ohm. Desmos Ohms form between two adjacent cells. Here's what I was referring to as titch buttons, two titch buttons coming together and sealing these two cells or making these two cells stick with each other. So the cytoplasmic portion of these buttons is attached to a network of intermediate filaments. So here in blue, we can see all these intermediate filaments. So just like in adherent junctions, the network of actin filaments basically was connected from one cell to another cell to the next cell. So there was a transcellular actin filament network. Here you can think of this. There is a transcellular network of these rope-like strings that are going from one cell to the next cell to the next cell via these structures called Desmos Ohms. And in Desmos Ohms, the sticky molecule again is the cadherin molecule. So here these are the blue ones. The blue lines are intermediate filaments, and these filaments have great tensile strength. So when we pull on our skin, it is basically we are applying tension, and this is resisted by these type of filaments, the intermediate filaments. So intermediate filaments attached to Desmos Ohms depend on the cell type. For example, in epithelial cells, the type of intermediate filament that is attached to Desmos Ohms is the keratin filament, which is type of an intermediate filament. In hard muscle cells, for example, the type of intermediate filaments that are linking different Desmos Ohms, that type of intermediate filament is called Desmon filament. Let's look at that little plaque or titch button in more detail, in a more magnified form. Here we have this dense material. We'll talk about what is in here later on. But basically, there are certain proteins which are hooking or linking these sticky molecules, these cadherin molecules, with the intermediate filaments. And they appear like dense plaque under the electron microscope. So this whole structure basically is the Desmos Ohm. Here you can see ore-like proteins. These are special type of cadherin molecules. These are the two plasma membranes of adjacent cells. And now let's look at a little bit more detail of this plaque, these different proteins. But before I move on to that, let me mention that these cadherin molecules are calcium dependent, homophilic. They use homophilic mechanism to attach with one another. Homophilic means two similar type of proteins, two same proteins can stick with each other, and they are dependent upon calcium, presence of calcium ion. Here again, as I mentioned, I'll show you a more magnified view of that particular Desmos Ohm. There are two type of, cadherin molecules that are present in Desmos Ohm. These special molecules are called desmoglein. The light green ore-like structure here is the desmoglein, and the dark green ore-like structure is the desmopollen. These proteins have three domains: extracellular domain, the intramembranous domain, and the cytoplasmic domain. The extracellular domain of these desmogleins and desmopollens, they stick with the desmogleins and desmopollens of the adjacent cells, and their cytoplasmic domains bind certain proteins, anchor proteins. For example, placoglobin, desmoplakin. These proteins basically are serving as linkers which link these sticky molecules with the intermediate filaments that we talked about. I would like to further mention here that placoglobin is also called gamma-catenin, which we have talked about catenins briefly in signaling. It's the same category of protein that basically is, when we talked about APC, we talked about them. Here, again, we have electron micrographs showing these darkly stained densities. You can see them here. And you can, if you, in the low mag, you can sort of imagine these filaments coming towards this dark, dense entity, which is the Desmos Ohm. And when we magnify the view, we see that this dark density here, these are the proteins, which I said, the anchor proteins, which are linking our sticky molecules, cadherin molecules, with the intermediate filaments. Here you can see that these intermediate filaments, they're going in towards this dark plaque, and which is between the adjacent cell membranes. There are these cadherin molecules that are sticking with each other. I would like to mention here something of general interest maybe. There's a disease called Pemphigus vulgaris. It's potentially fatal skin disease. Affected individuals basically in this disease make antibodies against one of their own desmosomal cadherin proteins. So cadherin proteins are very important, as we have seen, and if there's an antibody made against these proteins, they will not be able to function properly. As a result, these antibodies can disrupt the desmosomes that are holding skin epithelial cells, which are also referred to as keratinocytes, together, and we see that there's blistering of the skin and there's seepage of different fluids into the cavities where they should not be going. And this is basically how a person suffering from this disease looks like. So we have talked about desmosomes, how desmosomes of adjacent cells form these structures which are basically responsible for linking actin filaments, linking the intermediate filaments of two adjacent cells, and this network goes from one cell to the next cell to the next cell. So cells in a row, you can think of them having a single basically network of intermediate filaments, of course, that are linked together through the desmosomes, which have these cadherin proteins.